Because
Yes, ma'am, you can
I
start.
can see only the screen. I cannot see the audiences and anything. I can only see the my screen only.
No, you will not see the audience.
Yes, yes.
Shweta, ma'am, you can start. Live is started.
Okay, so
Okay,
thank you.
okay, yes.
Good evening, esteemed guest, respected faculty, dear students, and valued member of the Microbiology Society. A warm welcome to this enlightening online lecture organized by the Microbiology Society. I am Shweta Fadtare, honored to be the convener and your host for this session. We are thrilled to connect with all of you through this digital platform for an evening of knowledge and inspiration us all. Before we begin, please ensure your microphones are mute to avoid interruption. Let's delve into this exciting session. The Microbiology Society is a dynamic platform dedicated to advancing the field of microbiology through education, research, and collaboration. Under the visionary leadership of our president, Dr. A.M. Deshmukh sir, the society has been organizing impactful events to foster scientific curiosity and innovations. We are excited to have you all wish us today. It's my great pleasure to introduce our today's guest speakers, Dr. Avinash Raut sir, Assistant Professor in the Department of Microbiology at Yashwantrao Savan College of Science, Karad. Dr. Rao sir holds an PhD in microbiology along with set qualification in life sciences with 16 years of teaching experience at both undergraduate and postgraduate levels. He is a passionate educator and a researcher specializing in microbial ecology and extremophilisis. His innovative works on nitrogen fixing microorganism from lunar lakes has been published in 19 national and international journals, and he has been recognized as a reviewer for scientific reports by Nature Publication Group. Dr. Rao sir contributes also include organizing workshops and webinars, making him a true leader in the field. Without further delay, I invite Dr. Avinash Raut sir to take over and share his expertise on from Meteor to microbes, the non-symbiotic nitrogen fixers of Lunar Lake isolated at Yashwantrao Chavan College of Science, Karad. Dr. Raut sir, the virtual stage is yours. Please go ahead.
Thank you very much, ma'am. So the screen is visual to you. That the heading is visual to you from Meteor to Microbe, the non-symbiotic nitrogen fixers of Lonar Lake. We isolated at YCCS Karad, means Yashwantrao Savan College of Science, Karad. My research guide was Dr. Sham Bajekal, sir. Uh, this slide represents that I have modified the title. Actually, the title runs like this. This is my first title which I have submitted on 2012. Studies on aerobic non-symbiotic nitrogen fixing microorganism from littoral soil of Lonar. This was a specific title which we had designed for the synopsis as well as the final Viva presentation. And I personally didn't change any of the slides of my PhD Viva presentation because it was a memorable day for me completing my PhD degree, and that's why I hardly could change any of my slides. So only this slide have changed, only the title for the presentation purpose. Actually, this is the slide. So I should move forward to the introduction point of view that, as we all know, that nitrogen gas, which is abundantly found in the Earth's atmosphere, it is at 78% up to in the Earth's atmosphere, as well as essential components. It is essential component of all the life forms, as well as, but directly it cannot be utilized by all the life forms. It should be converted into soluble forms of nitrogen, which could be utilized by the living organisms. Biological nitrogen fixation, if you see, it is converted into partially utilizable form due to biological nitrogen fixation. But physical nitrogen fixation also has some significance, such as lightning and some cosmic clouds, which, when they are formed, they convert the nitrogen gas into ammonia. But significantly, biological nitrogen fixation plays a very important role on this earth. There are two types of nitrogen fixing bacteria: symbiotic nitrogen fixing bacteria as well as non-symbiotic nitrogen fixing bacteria. But I didn't try to isolate symbiotic nitrogen fixing bacteria because my source of sampling was very specific, and it was a world heritage, that is Lonar Lake, because it was characterized by soil alkalinity as well as water alkalinity as well as salinity. And the causes of soil alkalinity and salinity were quite different because of the closed basin lake structure of that huge meteorite impacted crater lake. And further, we move towards the remedy for the microorganisms which can be used against the saline soils as well as alkaline soils in our local areas, such as Sangli district as well as Kolhapur district, which were characterized by soil alkalinity as well as soil salinity because of tremendous flooding situations in 2005 to 2008. The next slide runs like this, that this is the image which we can see at the down part of the slide about the littoral soil of Lonar Lake, from where we had collected the soil samples. In total, we collected the samples of Lonar Lake's littoral soil from 2006 to 2011, and likewise we had to go through various constrictions while we were collecting the soil samples. Lonar Lake is characterized by alkaline as well as moderately saline lake. It is a very renowned lake in the Maharashtra as well as in the India. In India, there are only two types of meteorite impacted lakes. This is a lake which was formed because of a meteorite impact. Some say it has been formed 52,000 years ago. Some say it has been formed 1 lakh years ago. But various speculations are there. Various scientists visit the Lonar Lake in a year. Most of them are from NASA as well as from Japan, and many more scientists are visiting regarding to the geological point of view, zoological point of view, botanical point of view, as well as microbiological point of view. As my guide was very fascinating related to this lake, he told me to opt this particular source for isolation, and then the journey started in 2007. My provisional admission was initiated at 2001, 1st January 2007, and likewise it was started. So if you see the microbial diversity of Lonar Lake, many of the scientists have worked on it, and many microorganisms have been isolated from this holy lake. Because I call it holy because it was my workplace, and in Marathi we call it as a karma bhumi, and that's why I always admire this lake. And whenever I visit the northern Maharashtra region, I immediately my car goes towards that Lonar lake. So various types of microorganisms have been isolated from this lake, and you can see in the table that various references have been mentioned over there since 2000 since 1993 to 2011. Many scientists have worked on it, and likewise now also many people have worked on that particular ecosystem. Now, the aim of my PhD topic was to isolate and study. My topic was not so broad. The students who are working on their problem of PhD, don't be so exaggerated on your topic. You should be very specific in your topic, and I was also one of them, to isolate and study non-symbiotic nitrogen fixing microorganisms Only I had focused on bacteria as well as actinobacteria from this location, and possibly applicability of these microorganisms to saline and alkaline soils. This was my very primary study which I had initiated. You can see the very you can see the uh, view of Lonar Lake from the western region of this particular region. Now, what was my objective of current investigation? It was very initial because you can see that I have registered for PhD in 2007. So, and being a very I was not so good at particular the work. That's why my objectives are very specific to collect the literal science of Lonar Lake, to track the latitude and longitude of the each side using the GPS software. GPS software was available in my years. Enumeration, enrichment. This is a completely classical microbiological work which I had done in my PhD days. Okay, enumeration, enrichment, isolation of halotolerant and alkalophilic non-symbiotic nitrogen fixing bacteria from the soil samples. And initially, uh, the trend was initiated that you have to isolate the 6-ness rRNA analysis and do the phenotypic characterization. Many researchers and many uh, my well-wishers used to urge me to subject your bacterial isolates to 6-ness RNA analysis. I had initiated that particular thing, and I mentioned it in my objectives. Then to study the nitrogen fixing potential, which was my primary objective, and study on their plant growth promoting support potential.
Excuse me,
Yes,
Avinashi. Yes, this is Dr. Prasun.
yeah.
Avinashi, your slides are not moving, so audience are unable to see the slides.
My slides are not moving.
Yes, yes, it is still stuck at that part. Okay, now we can see some movement.
Really, but
You can also intermittently check the chat box.
can you see my slides now?
Yes, now you are at slide 8.
Yeah.
You continue, sir.
Can you see my slides?
Yes, slide
Yes.
number 8 is visible. Mm.
You cannot.
Sir, your slide is visible. You can continue now.
No. Just my slides are visible now.
Yes, yes. Now visible.
It's now visible
No.
now. Are they visible now?
Yes.
Okay. So the review of relevant literature which I had worked on. First point was nitrogen soil, symbiotic nitrogen fixing microorganisms, non-symbiotic nitrogen fixing microorganisms, and all those things which my guide has suggested to me. All those characters I had mentioned in my review of literature, and that's accordingly I went on extensively working on it, and I was successful. This is the can you see this view of Lonar Lake from the satellite image? Only say yes, because it would confirm me that I am on the right track. I am explaining it to you. Yes, Chetan ma'am.
Yes.
Chetan,
Sorry, sir. Sorry. You can continue.
can you see
Uh, no, sir. Your slide.
this slide? You cannot see.
No, no slide.
Now can you see?
Uh, no, sorry. Yes, now. Now see.
Now can you see the slide?
Yes, yes.
Yes.
Yes, sorry. Yes, sir.
Okay.
Continue.
Okay. Thank you very much.
Yeah, thank you.
So these were the sites which we had initiated to to collect the samples from, because our scrutiny committee had suggested to mention all those sites from where we had collected the slides, and likewise I mentioned all those slides using, at that time Google Earth was available. If you can memorize those things or put instead of Facebook and Google, instead of Google Maps, that Google Earth was available, and these images are from Google Earth, and you can see the images which we had uh, drawn from that particular spots from where we had collected the samples. This is the ecosystem of Lonar Lake, from where we can see that the lake is always covered with salt on the surface of the soil. Now, the materials and methods were quite specific, to the point, as suggested by the RAC committee as well as the RRC committee of our Shivaji University Kolhapur. They were very simple: collection of soil samples, chemical analysis of soil samples, enumeration, enrichment, and all those things. You can read all those things. Nitrogen fixing potential of the isolates, effect of environmental factors, plant growth promoting support analysis by hot studies, and tolerance of those isolates to various agricultural pesticides which were used commonly in our Sangli district as well as in Kolhapur district. Okay. Now, this quote is very important in our point of view, because Alan Bull in 1992, the team of Alan Bull, they had mentioned that biodiversity is a source of innovation in biotechnology. Biotechnology is incomplete because unless and until you study the biodiversity, and that
Sir,
only we had studied.
sir, slide are not moving. Yes.
You can see here.
Yeah.
So, okay, I will mention here only like this. I am in a, I have. I will not click the slideshow button. Okay.
Okay.
Okay. So biodiversity is a source of innovation in biotechnology, that was mentioned by Alan Bull in 1992, and accordingly we started our journey of biodiversity of Lonar Lake. So these are the. Let's come to the next point, that is the results and discussion point of view, which shows the sites from where we collected the samples. Is this slide visible to all of you?
Yes, sir.
Yes. Okay. Thank you. So these are the soil characters which we had mentioned, and which we saw and mentioned according to the coordinates of GPS in that area. These are the chemical characteristics of the Lonar Lake soil samples which we performed in our laboratory. Which you can see that, you can see this point, which I have making it bold, that carbonates and bicarbonates are abundant in this Lonar Lake soil as well as in water sample. And likewise, you can see that the isolates codes which we had given to the bacterial as well as actinobacterial isolates were running like this. The isolate codes which we had mentioned here, as well as the accession numbers of the genomic 6-ness RNA analysis which we had conducted, and the percentage similarity of those isolates with the available databases. Next, according to the genus and species, we had, according to the genus and species which is available in Burgess Manual of Systematic Bacteriology, we classified those microorganisms which we had found in Lonar Lake. And these are the colony characteristics which we had found. Gamma proteobacteria, alpha proteobacteria, then bacillus species, and actinobacteria species. These are the biochemical characteristics of all those isolates according to the previous classification. I'm moving the slides one by one, which we had mentioned it in our thesis. This is the graphical representation of the bacterial as well as actinobacterial isolates which we had obtained from the various sites according to the directions: east, southeast, southwest, south, northwest, north, west. Okay, accordingly, which type of isolates we had obtained, that is represented in this bar diagram. These are the phylogenetic positions of the bacterial isolates which we conducted in the phylogenetic analysis. These are the actinobacterial isolates. And total number of and the variety of nitrogen fixing bacteria which we had obtained in the littoral zone around the Lonar Lake. Means in east we found these types of microorganisms. In the stream of that Lonar Lake we had found these types of microorganisms. From southeast region we had found these types of microorganisms. Likewise, all those uh, directions we had mentioned that which type of microorganisms of nitrogen fixing bacteria, non-symbiotic nitrogen fixing bacteria, we are obtained from various types of sites. And this is the nitrogen fixing ability and the potential of the isolates to fix nitrogen, total nitrogen content conducted by the traditional nitrogen content method, that is Gelda's nitrogen fixing method, as well as acetylene reduction assay method. And quality control which we had used was a Azotobacter crucocum species, which is a very potent nitrogen fixing bacterial at pH 7. Okay, and accordingly, these one thing I want to mention is that all the other above isolates, they were cultivated as well as grown, as well as the nitrogen fixing ability was studied at pH 10 and above. And positive control which we had used, because azotobacter is not tolerant. When we carried out the study, azotobacter could not grow at alkaline pH. Now it may be able to grow, but I don't know about today. But at that time when we conducted the study, azotobacter crucocum was not able to cultivate at alkaline pH, and that's why we cultivated it at 7 pH, and all those studies were carried out using that pH only, and those results are mentioned here. They are related to that. Comparative analysis was related to azotobacter, which is a potent non-symbiotic nitrogen fixing bacteria, and the other nitrogen fixing bacteria which we had obtained from Lonar Lake. This is the environmental factors of nitrogen fixing bacteria which were obtained from Lonar Lake. Effect of pH, effect of temperature, effect of NaCl, means the salt which we had used in our project work was NaCl. That's why only we concentrated on NaCl. It doesn't mean that in Lonar Lake only NaCl is present. Many other salts may be present, but we had focused on only NaCl in our project. And these are the growth parameters and the nitrogen content which we obtained when the plants were uprooted from the pots and analyzed 2 months after the inoculation. These are the pot studies studies results. So you can see in this photograph that the positive control which we had used was Azotobacter rubocum, and in the left side you can see in the first photograph you can see uninoculated control, and other all those things are from the isolates which we had inoculated. The plant which we used was Triticum aestivum, that is wheat plant. Okay, because that was the only plant which we found it suitable, because wheat is consumed togetherly throughout India on a very large scale after rice. That's why we had selected the same plant, as well as we had also conducted the analysis to see whether these bacteria are resistant or susceptible to various herbicide as well as insecticides or pesticide concentrations. The Cartap hydrochloride was used by us at that time, and the results are mentioned over here in this slide. This is about the pesticide methomyl. This is a qualitative analysis and the various concentrations which we had used. Very minutely we had used the isolate concentration, so that we can see whether it is showing growth or not. This is the pesticide Folidol, which was very familiar to all of us. You can see that at 4% concentration, many of the isolates could not grow at that particular concentration. If you see that, in the summary and conclusion point of view, alkaline nature of littoral soil of Lonar Lake was confirmed. Average pH of that lake was 9, of that not lake, of that littoral soil was 9.57, and the carbonate and bicarbonate level was 1.03 mg per liter. As well as aerobic non-symbiotic nitrogen fixing bacterial flora was significantly high, as well as highest on the eastern bank, where the littoral zone was the biggest. Littoral zone in the eastern region is very biggest, because I don't know why, but the littoral zone which we found in 4 of the times of sampling was very high in the eastern side. And we had found 22 distinct species of bacterial as well as antibacterial, actinobacterial species, as well as the 5 classes of bacteria were isolated by us at our department, that is Aquifici, alpha proteobacteria, gamma proteobacteria, firmicutes, that is bacilli, as well as actinobacteria, according to the Burgess Manual of Systematic Bacteriology. Then the distribution around the lake, you can see that total number plus diversity is equal to highest in the east, followed by the southwest, northwest, as well as the southeast region. This topic of my PhD was totally regarding to the ecological point of view of littoral samples of Lonar Lake to isolate only nitrogen, non-symbiotic nitrogen fixing bacteria from that particular region. If you can see that alpha proteobacteria was predominantly found in the eastern region, actinobacteria were found in the southern region, bacilli were found dominant in the western region, and the Aquifici family was found in the only in the south and the northwest region. Phenotypic characters were also studied, and confirmed nitrogen fixing ability was confirmed by Geldal methods as well as acid reduction assay. Influence of pH, temperature, and salt were conducted by the nitrogen fixing potential. Best nitrogen fixing conditions of the nitrogen fixing bacteria from Lonar Lake was ranging from 10 to 12 pH range, as well as the mesophilic temperature was 35 to 40 degrees Celsius. Moderate salinity was 1 to 5%. As compared to all those analyses which we had conducted and all, when we came to the conclusion that Spingomonas azotophagians was the best nitrogen fixing bacteria, but none was equal to Azotobacter crucocum, which can fix nitrogen at neutral pH. Slackia exugoa was one of the actinomycet, which was fixing quite significant nitrogen at alkaline pH, but it was actinobacter. We had also conducted a field study in soil-prone regions of Sangli district, where the soil is saline as well as alkaline, and we found very significant results. But that study was initiated after my completion of my PhD. So these are the conclusions which we found in our study. And these are the selected references which we had used during our project work. So these are some of my significant publications which we had conducted during the PhD work. Thank you very much.
Thank you, sir. Thank you for such a ins- insightful lecture. Your expertise has truly enriched our understanding of from Meteor to microbes, the non-symbiotic nitrogen fixers of lunar lake. We now have some times for a question answer session.
Yeah.
So please submit your question in the chat box. Mm. If no questions, it seems we are all still processing the wealth of knowledge shared. If any questions come up later, you can reach out through the Microbiology Society group. To express our heartfelt gratitude for Dr. Raut's sir exceptional presentation, we extend a virtual token of appreciation of behalf of the Microbiology Society. Your insights have inspired us all, and we are deeply grateful for your time and expertise. It is now my pleasure to invite Dr. M. Deshmukh sir, President of Microbiology Society, to share his thoughts and address our virtual gathering. Sir, please take the floor. Deshmukh sir. Okay, I think sir is busy. So, as we draw this enlightening session to a close, I extend my heartfelt gratitude to everyone who made this online lecture a success. A special thank you to our distinguished guest speaker, Dr. Avinash Raut sir, for sharing his profound knowledge with us. We are deeply grateful to Dr. A. M. Deshmukh sir, President of the Microbiology Society, for his guidance and support. My sincere thank to the organizing team and technical coordinators for ensure a seamless virtual experience. And finally, a big thank you to all of you, our wonderful virtual audience, for joining us from across the world and making these events vibrant with your participation. Please keep engaging with the Microbiology Society for more such enriching events. With that, I declare this online lecture conclude. Wish you all a wonderful evening. Thank you. Thank you so much.
Excuse me, sir. Rajnish Patil this side. So I
Yeah.
want to talk to you if I get your contact number.
Yes. Shall I share it in my chat box?
Yes, 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 of course, I will know.
Yeah.
And I request to Microbiological Society of India, please add me in this group. I was
Uh,
not in group presently.
yes, sir. Yes.
So I had mentioned it here.
Yes, sir. Yes,
So Rajneesh Bhatia sir, where are you from?
I'm from Haryana, sir.
Isar?
Bhiwani.
Okay, okay, Bhiwan.
I was RD head in CSSHU in biofertilizer department, and after my resignation, I start my own startup, and I'm working on them.
Okay, really good. It's a good startup.
Yes, sir. Thank you.
Yeah.
Sir,
I'll talk to you on the call whenever you're free.
Yes,
Okay.
yes. I have
sir,
Okay.
shared my
please.
number. You can call me anytime.
Yes, sir. Yes, sir. I just noted.
Yeah, okay, fine. Nice to meeting you. And
Thank
all
you, sir.
the I thank one and all from the Microbiology Society of India who gave me opportunity to talk on my PhD topic. Thank you very much.
Yes, sir.
So
Yes.
shall I leave now?
Yes, sir. Yes. Thank you so
Thank you very
much.
much. Okay.
Yeah.
And ma'am, please add me in this group.
Sir, please share your number.
Just a minute.
Okay, sir. I will add you.
Okay, ma'am. Okay.
Okay,
Thank
sir.
you.
Okay.
Yeah.
Good
Thank you
night to
so
all of you.
much, everyone.
Good night.
Yeah.